First one, C3 plants. Generally grown in temperate and tropical areas. C4 plants found in tropical and subtropical areas. Then next one, in C3 plants, chlorophyll present only in mesophyll cells. C3 plants, chlorophyll present only in mesophyll cells. C4 plants, chlorophyll present in both mesophyll and also bundle sheath cells. Mesophyll and bundle sheath cells. C3 plants, only one type of chloroplast. Only one type of chloroplast is present in C3 plants. C4 plants, two types of chloroplast. C4 plants, two types of chloroplast is present. How two types means? One is mesophyll chloroplast. In mesophyll chloroplast, thylakoid membranes organized into grana called a granule chloroplast. Bundle sheath chloroplast, thylakoids present but are not organized into chloroplast. So, mesophyll chloroplast is called a granule and bundle sheath chloroplast is called agranule. So, rightly it is called chloroplast dimorphism. So, chloroplast dimorphism exhibited by C4 plants. C3 plants, only one type of chloroplast present. Then C3 plants does not show Kranz anatomy. There is no Kranz anatomy. But C4 plants, they shows Kranz anatomy. Kranz is German word. English it is called a wreath. Rightly, the meaning of wreath is garland of flower. So, in C4 plants, Kranz anatomy is present. C3 plants, Kranz anatomy absent. Then C3 plants, only one carbon dioxide fixator is present, that is ribulose bisphosphate. Rightly, it is pentose, 5 carbon compound. In C4 plants, 2 carbon dioxide fixators present. One is triose, that is phosphoenol pyruvic acid. Second one, ribulose bisphosphate, that is pentose, 5 carbon compound. And in C4 plants, first carbon fixator is PEP. It fixes carbon in mesophyll cells. Second carbon dioxide fixator that is RUBP. It fixes carbon in bundle sheet cells. But C3, only one carbon dioxide fixator is present. First product in C3 plants, phosphoglyceric acid. So it is 3 carbon compound. That is why that cycle is called C3 cycle, and plants showing C3 cycle are called as C3 plants. In C4 plants, first formed compound is oxaloacetic acid. That oxaloacetic acid is 4 carbon compound. So in C4 plants, first formed 4 carbon compound, that is oxaloacetic acid. So these are called C4 cycle, and plants showing C4 cycle are called C4 plants. Then in these C3 plants, they exhibit photorespiration. There is photorespiration in C3 plants. Almost absent or no photorespiration in C4 plants. No photorespiration in C4 plants. Then C3 plants. C3 plants, they shows carbon dioxide compensation point. C3 plants, but negligible or almost absent this carbon dioxide compensation point. No carbon dioxide compensation point in C4 plants. And C4 plants does not show efficient carbon dioxide fixation. Then C4 plants, they show sufficient carbon dioxide fixation, because in C4 plants that efficient carbon dioxide fixator is present in these C4 plants. Then next one, C4 plants, optimum temperature in C3 plants 15 to 25 degrees. Optimum temperature for C4 plants 30 to 45 degrees. 30 to 45 degrees optimum temperature for C4 plants, 15 to 25 in that C3 plants. So next one is photorespiration. Here photorespiration is also called C2 cycle or PCO, photosynthetic carbon oxidative cycle, or also called glycolate metabolism. Also called glycolate metabolism. Then what is photorespiration and how photorespiration takes place? In which plants photorespiration occurs? These are points. First point, this photorespiration occur in C3 plants. So photorespiration occur in C3 plants. Actually, first step in a C3 cycle is carboxylation. Normally 1,5-ribulose bisphosphate, this 5 carbon compound, it fixes 1 carbon dioxide and forms 2 molecules of phosphoglyceric acid. So first step is carboxylation. RUBP fixes carbon and forms 2 molecules of PGA. This reaction is catalyzed by Rubisco. Rubisco means RUBP carboxyl oxygenase, and actually this enzyme has 2 active sites. One is for carbon dioxide, and second is for oxygen. And this enzyme, it shows more affinity with carbon dioxide. So normal condition, if carbon dioxide amount is more, RUBP fix carbon dioxide and it forms 2 molecules of phosphoglyceric acid. That is normal condition. But point is, if oxygen amount is more, that enzyme acts as oxygenase. Once this enzyme forms oxygenase, instead of carbon dioxide, this enzyme fixes oxygen. Once, if this enzyme fixes oxygen, it has only 5 carbons and it forms 1 molecule of phosphoglyceric acid and 1 molecule of phosphoglycolic acid. And this phosphoglyceric acid participates in C3 cycle. Then this phosphoglycolic acid or phosphoglycolate, in this phosphoglycolate, dephosphorylation takes place and this phosphoglycolic acid is converted into glycolic acid. And a main substrate for photorespiration is this glycolate or glycolic acid. Hence, this cycle is called glycolate metabolism. Hence, this cycle is called glycolate metabolism. Then, up to glycolic acid, these reactions occur in chloroplast. Who discovered chloroplast? So, chloroplast discovered by Sachs. And the term chloroplast was given by that Schimper and discovered by Sachs. Then, from chloroplast, this glycolic acid enter into peroxisome. From chloroplast, glycolic acid enter into peroxisomes. What is peroxisome or why that name is given? Peroxisome means actually it contains peroxide oxidizing enzymes. And these peroxisomes were discovered by Talbot. Then, once glycolic acid enter into peroxisome, this glycolic acid undergoes oxidation and forms glyoxylic acid. Then, co-product of glyoxylic acid is hydrogen peroxide. Then, this peroxisome contains catalase enzyme. This catalase enzyme, it breaks that hydrogen peroxide into water and oxygen. Afterwards, this glyoxylic acid, it accepts an amino group and it forms amino acid glycine. Then, from peroxisome, this glycine enter into mitochondria. From peroxisome, glycine enter into mitochondria. Here, already this mitochondrial matrix contains some amino acids because in matrix Krebs cycle takes place and intermediate product of Krebs cycle is alpha ketoglutaric acid It is precursor for amino acids. So, this matrix contains many amino acids. Along with many amino acids, this matrix contains glycine. So, in mitochondria, these two glycine molecules undergoes oxidative decarboxylation. And forms another amino acid, that is what means serine. Actually, respiration. What is respiration? Respiration means oxygen fixed and a carbon dioxide is released. Here, this oxygen fixation, it occurs in chloroplast, but carbon dioxide is released in mitochondria. But this step, this intaking of oxygen and liberation of carbon dioxide, it occurs only in presence of light. Hence, it is called photorespiration. And first form product is phosphoglycolic acid. It has 2 carbons, so it is called C2 cycle. And main substrate for photorespiration, glycolate. So, it is called glycolate metabolism. This photorespiration differs from normal respiration. How means? Photorespiration occur in all types of respiration occur in all types of cells. Photorespiration occur in green cells and normal respiration occur in all times, but photorespiration occur only in daytime. Then after formation of serine in metabolism in a mitochondria, by metabolism, this serine enter into peroxisome. In peroxisome, this serine by deamination forms hydroxypyruvate. Then this hydroxypyruvate reduced to glycerate. Finally, this glycerate enter into chloroplast, reacts with ATP and forms phosphoglyceric acid and participate in that C3 cycle. So, photorespiration occur in 3 cell organelles: chloroplast, peroxisome and mitochondria. Then finally, it is wasteful process, because in respiration ATP and NADH2 formed, but instead of formation this ATP and NADH2 utilized. So, it is wasteful process. It occur only in C3 plants, but not in C4 plants, because C4 plants contains efficient carbon dioxide fixator. C4 plants, it contains efficient carbon dioxide fixator. This efficient carbon dioxide fixator always fix that a carbon. So, always in C4 plants, intracellular carbon dioxide concentration is increased, and always this carbon dioxide concentration is more than that oxygen. So, no chance of photorespiration in C4 plants. Only in C3 plants this photorespiration takes place, because C3 plant does not contains efficient carbon dioxide fixator, that is what means that a PEP. That is why this photorespiration occur in C3 plants and it reduce that photosynthesis and previously it was first reported by Warburg. So, photorespiration is also called Warburg effect. Next one, factors. Factors influencing that rate of photosynthesis. Actually, photosynthesis is influenced by both external and also internal factors. External and internal factors. External factors includes light, carbon dioxide, temperature, oxygen and water. These are external factors. Internal factors. Internal factors includes chlorophyll content, then internal structure of leaf, then accumulation of end products. Accumulation of end products. So, first one, light. Light, it influences photosynthesis in three ways. One is quality, second one intensity, and third one duration. Quality, intensity and a duration. Quality. Quality means here ultimate source of energy for photosynthesis. So, light means sunlight. So, sunlight is source of energy for photosynthesis. Then light on spectrum, light exhibit in two forms, visible and invisible parts. Then visible range of light generally 390 to 760 nanometers. This visible range of light 390 to 760 nanometers. Rightly, it is called a PAR. PAR means photosynthetic active radiation, and it exhibits 7 different colors. Then light shows actually wave and a particulate nature. That particulate nature is expressed in the form of photon or quanta. Rightly, energy in each photon is inversely proportional to energy in each photon is inversely proportional to that quanta. So, point is blue light is more energetic than red light. Blue light is more energetic than red light, and point is maximum photosynthesis occur in a red light, next in blue light, and least in a green light. So, it is quality. Then next one, intensity. Here some plants, they grown under high light intensity. If plants grown under high light intensity, called heliophytes. Some plants, they grown under low light intensity, called cyophytes. Generally, with the increase of light intensity, that the photosynthetic rate is increased. With the increase of light intensity, photosynthetic rate is increased, but point at high light intensity, chlorophyll structure is destroyed. That is called photooxidation, or it is called photo bleaching, or it is called solarization. So, with the increase of light intensity, photosynthetic rate is increased, but at high light intensity, that chlorophyll structure is destroyed, called photooxidation, photobleaching, or solarization. To protect that reaction center or chlorophyll from this high light intensity, around reaction center, accessory pigments present. Which pigments? Accessory pigments present. And in higher plants, these accessory pigments contains chlorophylls and carotenoids. In higher plants. But particularly in blue-green alga, these accessory pigments includes chlorophylls, then carotenoids, and also phycobilins. These phycobilins are exclusive algal pigments found in blue-green alga. They are water-soluble photosynthetic pigments and also act as accessory pigments. So, these are quality, intensity, and third one, duration. Duration means, for example, if you take two leaves, one leaf is exposed to continuous light. One leaf is exposed to continuous light, and second leaf exposed to discontinuous light or flashes of light. Here it is total continuous light. Then second one, discontinuous light, means this light period is interrupted by dark. Then in which leaf more photosynthesis takes place, means the leaf which is exposed discontinuously or flashes of light. Why? Means rate of photosynthesis directly proportional to rate of translocation of food. So, if light is continuous, more food materials are accumulated. If more food materials accumulated, they blocks active sites of enzymes. That is called feedback inhibition. So, in continuous light, that is why photosynthetic rate is decreased. In flashes of light, in that gap, that food materials translocated to storage pods. So, there is no feedback inhibition. That is why compared to continuous, in flashes of light, more photosynthesis takes place. So, it is about first one, quality, intensity, and duration. Second one, carbon dioxide. Actually, carbon dioxide also influences that photosynthesis, and carbon dioxide is one of the raw material for photosynthesis. Actually, in dark reaction, that carbon dioxide reduced to carbohydrates. So, carbon dioxide is also essential for photosynthesis. Then point is, this atmosphere contains 0.03% or 0.04% of carbon dioxide. 0.03 by 100 into million forms 300 ppm of carbon dioxide. 0.04 by 100 into million forms 400 ppm of carbon dioxide. So, general atmosphere contains 0.03% to 0.04%. It means 300 to 400 ppm. With the increase of carbon dioxide up to 500 ppm, that photosynthetic rate is increased. Then beyond that this carbon dioxide become toxic to that chloroplast, particularly guard cells affected, so stomata closed, so photosynthetic rate is decreased. It is one of the factor. Second one, beyond that limit, it becomes limiting factor and also reduce that photosynthesis. Then actually C3 plants and C4 plants, C3 and C4 plants at a low light intensity, both plants do not respond to that carbon dioxide. Here C3 plants and C4 plants. C4 plants saturated at 360 ppm of carbon dioxide, C4 plants. C3 plants, they saturated at 450 ppm. So, compared to C4, C3 plants saturated at 450 ppm. That is why some of the C3 plants like pepper and tomato grown under high carbon dioxide concentration for more productivity. This is carbon dioxide. Then next one, temperature. Next one is temperature. Here optimum temperature for C3 plants 15 to 25 degrees. For C4 plants it is 30 to 40 or 45 degrees. So, compared to C3, C4 plants carry out photosynthesis even at 45 degrees. These are optimum temperature, but photosynthetic rate is decreased at low and high temperature. At low temperature, enzymes are inactive. At high temperature, enzymes are denatured. That is why at low and high temperature, photosynthetic rate is decreased, but optimum temperature, they shows more photosynthesis. And some plants like coniferous plants, gymnosperms, these coniferous plants, they carry out photosynthesis even at -6 degrees. And lichens, they carry out photosynthesis even at -20 degrees. Opentia is xerophyte, carry out photosynthesis at 55 degrees. And some algal members, hot springs, these hot springs carry out photosynthesis at 70 degrees. So, it is about temperature. The next one, oxygen. Next one is oxygen. Your point, oxygen is not used in photosynthesis. Instead of utilization, this oxygen released during photosynthesis. But this oxygen effect on photosynthesis, particularly this inhibitory effect of oxygen on photosynthesis. Inhibitory effect of oxygen on photosynthesis in C3 plants, first reported by Warburg. So, it is called Warburg effect, and it is nothing but photorespiration. So, already I discussed, when oxygen concentration is increased, that Rubisco, that carboxylase enzyme is converted into oxygenase. Once that enzyme becomes oxygenase, instead of carbon dioxide, that RUBP fix oxygen. So, photorespiration takes place and photosynthetic rate is decreased. Then next one, water. Water is also like carbon dioxide, even water is also one of the raw material for photosynthesis, and particular in plants, this water act as electron donor. So, water act as electron donor, then that oxygen released into atmosphere comes from this water only, and point these released electrons and protons accepted by NADP and forms NADPH plus H plus. Actually, this Hill scientist, Hill, could isolated chloroplast release oxygen in presence of proton acceptor. So, Hill reported isolated chloroplast could release oxygen in presence of proton acceptor, and he used some artificial proton acceptors, that is potassium ferric oxalate. That potassium ferric oxalate is called Hill oxidant, and natural proton acceptor is NADP. So, water is essential for photosynthesis because it acts as electron donor. Second one, other importance of water. Water maintains turgidity of cells, and most of the enzymes are hydrolytic. They are active only in presence of water. So, these are external factors: light, carbon dioxide, temperature, oxygen and water. Internal factors. These internal factors includes chlorophyll content, then structure of leaf, and end product accumulation. End product accumulation. So, first one, chlorophyll content, rate of photosynthesis proportional to that chlorophyll content. So, young leaf shows more chlorophyll. So, in young leaves, that more photosynthesis takes place. The matured and old leaves means senescent leaves. Senescent leaves, loss of chlorophyll takes place. So, photosynthetic rate is decreased. Particularly plants grown under dark shows etiolation. Etiolation means that a plant become pale and thin, long. It is due to elongation of internodes. That is called etiolation. When this etiolation occurs means in the absence of that chlorophyll, means when plant kept in dark. Then structure of leaf. Structure of leaf also influences rate of photosynthesis. How means? Presence of thick cuticle, thick walled epidermis, number and a distribution of stomata, even mesophyll content also influences the rate of photosynthesis. Then final one, End product accumulation. End product accumulation means if more end products are formed, they blocks active sites of enzymes. So, photosynthetic rate is decreased. Rightly it is called feedback inhibition, or it is called end product inhibition. This feedback or end product inhibition seen only in C3 plants, but not in C4 plants, because in C4 plants bundle sheath cells help in immediate translocation of food. That is why we cannot see this process in a C4, only in C3 plants. This feedback inhibition can be seen. Now, last one, law of limiting factor. Law of limiting factor proposed by Blackman. Previously it is called law of minimum. Law of minimum proposed by Liebig. What is law of limiting factor means? If any metabolism is conditioned by many external factors, its rate depends on least available factor called limiting factor. Then generally limiting factors for photosynthesis, carbon dioxide and light, but most common limiting factor is carbon dioxide, rare case only in cyophytes, light become limiting factor. Here this graph shows carbon dioxide becomes limiting factor. Carbon dioxide become limiting factor. How means? If light intensity increased beyond 200 units, that rate of photosynthesis did not increase, it becomes constant. So, for the formation of 6-RUBP in regeneration cycle, how many G3P molecules are used? That is question. Actually, 6-RUBP fix 6 CO2. 6-RUBP fix 6 CO2, then 12 G3P are formed. Out of 12, 2 G3P export into cytosol. For what? Glucose formation. So, how many G3P left? 10 G3P left. This 10 G3P used for regeneration of 6-RUBP. So, answer is 10 G3P. Next, for regeneration of, for formation of the 6-RUBP in regeneration cycle, how many G3P and DHAP are separately used for formation of 6-RUBP in regeneration? How many G3P and DHAP are separately used? Total 10 G3P used, but out of 10, 6 G3P directly used for regeneration of 6 RUBP and 4 molecules of DHAP. Out of 10, 4 molecules of G3P isomerized to 4 DHAP and remaining 6 G3P directly used. So, answer is 4 DHAP and 6 G3P. Then next question, what is a sequence? What is the sequence of carbon? A carbon number formation in that Kelvin cycle, means triose, tetrose, pentose, like that. So, first triose, PGA, 1,3-bisphosphoglyceric acid and G3P. After that, G3P and DHAP fuse to form fructose. So, next 6 carbon. After that, fructose monophosphate reacts with G3P, forms 4 carbon compound and 5 carbon compound. Next, after that, 4 carbon and 3 carbon fuse to form 7 carbon. After that, 7 and 3 carbon fuse to form 2 5 carbon compounds. So, answer is this sequence C3, 6, 4 carbon, 5 carbon, 7 carbon and 5 carbon. Next question, what is the ratio of carbon dioxide fixed to G3P molecules are formed? G3P molecules formed. So, generally 6 RUBP fix 6 carbon dioxide, then forms 12 PGA, DPGA and 12 G3P. So, answer is 6 is to 12, means 1 is to 2. Next, in Kelvin cycle, different carbon compounds are formed How many of them? Contains 2 phosphates, phosphate groups. Groups 3 carbon, 4 carbon, 5 carbon, 6 carbon, 7 carbon. So, 3 carbon, first PGA, then it forms 1,3-bisphosphoglyceric acid. So, it contains 2 phosphate, third carbon, it has 2 phosphate groups. So, answer 1, second on 4 carbon compound, only 1 phosphate is formed, erythrose 4 phosphate, no. 5 carbon compound, ribulose 1,5 bisphosphate. Then 6 carbon compound, fructose 1,6 bisphosphate. Then 7 carbon compound, pseudoeptulose bisphosphate. So answer is 1, 3, 4, 5. These 3 carbon, 5 carbon, 6 carbon, 7 carbon. These 4 contains biphosphate groups and also monophosphate groups. Next question. If chlorella cells exposed to red light of longer wavelength 36 photons followed by or simultaneously exposed to 20 photons of red light of shorter wavelength. How many? 20 photons. By cyclic and non-cyclic. By cyclic and non-cyclic, how many ATP and NADPH are formed? So, longer wavelength 36 photons, shorter wavelength 20 photons. So, for non-cyclic 20 photons of shorter, 20 photons of longer. It means 20 Z schemes takes place. These 20 Z schemes, it gives 20 ATP and 20 NADPH. 20 Z schemes gives 20 ATP and 10 NADPH. So, that 36 photons of longer wavelength among the 20 photons used in Z scheme. So, how many photons of light left means 16 photons of light. 16 photons of light means 16 electrons. 16 photons of light means 16 electrons. 16 electrons means 32 protons. 32 protons means 30 protons. These 30 protons gives 10 ATP. Extra how many? 2 protons. These 2 protons cannot give ATP. So, that cyclic it gives 10 ATP, non-cyclic it gives 20 ATP. So, total 30 ATP and how many NADPH means 10 NADPH. Hill and Bendall, they proposed flow of electrons in a non-cyclic, that Z scheme, cyclic electron transport, discovered by Frankel in rhodospirillum, Frankel in rhodospirillum. Non-cyclic electron transport, discovered by Arnon. Chemi-osmotic hypothesis. Chemi-osmotic hypothesis proposed by Peter Mitchell. It is applicable to both photosynthesis and respiration. Photosynthesis and respiration. Chemiosmotic hypothesis. Then oxygen released during photosynthesis comes from water. was experimentally proved by Ribbon and Kamen, Ribbon and Kamen. And during photolysis of water, both the macro and micro elements are involved. Macro element involved in photolysis of water, calcium. Micro element involved in photolysis of water, manganese and chlorine. And amino acid, which transfers electrons from lumen to PS2, tyrosine. Then Emerson exposed chlorella cells first to red light of longer wavelength. Then photosynthetic rate is decreased. It is called Emerson effect, or it is called red drop. In second experiment, he exposed chlorella cells to red light of longer and also shorter wavelength. Then photosynthetic rate is increased. That is called Emerson enhancement effect. Based on this, Emerson reported that two photosystems involved in photosynthesis. One is photosystem 1 and second one is photosystem 2. Photosystem 1 and 2. In eukaryotic cells, in eukaryotes, photosynthetic pigments present in chloroplast, but in prokaryotes, like a blue-green alga, photosynthetic pigments present in chromatophores. Then, in blue-green alga, main photosynthetic pigment, chlorophyll A. It means only chlorophyll A is present, that a B, C, D, E absent. Then, accessory pigments, it includes carotenoids and also phycobilins. Carotenoids and phycobilins. These phycobilins, exclusive algal pigments, present in blue-green alga and red alga. Then, structural element for chlorophyll A, chlorophylls, magnesium and iron is not a structural, but it is essential for chlorophyll formation. Then, light compensation point. Light compensation point means point at which carbon dioxide released by respiration absorbed by that photosynthesis, called a light compensation point. The next one, carbon dioxide compensation point, point at which that photosynthesis just compensates that respiration. It's called a carbon dioxide compensation point. Generally, C3 plants shows high compensation point and C4 plants negligible or almost absent.